So the paper we're diving into today,
Okay.
it describes the very first spatial transcriptomic atlas of Atlantic salmon skin.
Yeah, using this technique called Visium Spatial Gene Expression.
Okay, hold on. Visium Spatial Gene Expression.
Yeah. I
What?
mean, it's a mouthful.
What is that? Break that down for me.
Sure. So basically it's like,
Like for people who aren't scientists.
yeah.
Who aren't
Yeah,
geneticists.
exactly. So like traditional transcriptomics, it gives you a list of all the genes that are active in a tissue sample, right?
Okay.
But spatial transcriptomics takes it a step further, and it tells you where those genes are active within the tissue.
So it's
like,
I was going to say,
go ahead.
it's like a Google map for genes.
Exactly.
But instead of streets and buildings,
Yeah.
we're looking at, specific locations within the fish's skin.
Yes, exactly. You're zooming in and seeing, what's happening at a really granular level.
I'm curious why they chose Atlantic salmon specifically for this study.
Well,
Like, is it just because they're popular to eat?
I mean, that's definitely part of it, right? Atlantic salmon is a huge part of the aquaculture industry,
Right.
but they actually face a lot of health challenges.
Really?
Yeah, a lot of them are related to skin health because their skin is constantly exposed to the environment, right?
Makes sense.
So they're vulnerable to parasites and bacteria and all sorts of stressors.
Oh, so that's where understanding the skin's genetic activity comes in.
Exactly.
It could be the key to protecting these fish better.
Absolutely. If we know which genes are involved in immune responses, wound healing, stress tolerance,
Yeah.
we can potentially develop more targeted treatments, even breeding programs to make them tougher.
Okay, so they use this technique called Visium Spatial Gene Expression.
Right.
Can you walk us through that?
Yeah, so basically with traditional transcriptomics, you get like a parts list, right?
Okay.
You know what genes are there, but you don't know where they are or how they fit together.
Yeah.
Spatial transcriptomics is like getting the assembly instructions too.
So it's like the difference between having all the ingredients for a cake
Yeah,
versus actually knowing how to put those ingredients together to make the cake.
exactly. You got it. And that level of detail is super important because different areas of the skin have different jobs.
Right. And speaking of different areas of the skin,
Yeah.
the paper mentions it was tricky to get good quality skin sections from the fish.
Oh, it was a challenge for sure.
Why was that so hard?
Fish skin is delicate,
Okay.
And it's really hard to get those clean slices you need for this kind of analysis.
Right.
They had to modify the standard protocol, like they used a special embedding technique to stabilize the tissue during freezing and slicing.
So it's like, it's kind of like,
Like trying to slice a grape
yeah,
without it rolling away.
exactly. You need something to hold it steady.
Okay, so what did they find? What stood out to you in terms of which parts of the skin were most active?
Well, one thing they discovered was that the epithelial tissue,
Okay.
the outermost layer of the skin, had the highest mRNA count.
Which makes sense because that's the part that's directly exposed to the environment.
Right, it's the first line of defense. But here's the interesting part.
Okay.
The transcriptomy of the epithelial tissue was pretty much the same across different parts of the fish's body.
Really?
Yeah, so like whether you're looking at the skin near the head, the tail, or the belly, the genes that are active in that outer layer are pretty consistent.
Fascinating. So like a uniform barrier all over.
Exactly.
But the deeper layers of the skin.
Yeah, the dermal tissue,
Yeah.
that was different.
Yeah, that showed more variation in gene expression depending on the location.
Interesting. Okay, so why would that be?
Well, it probably reflects the different functions of the skin in different areas of the body,
Okay, I see. So like maybe tougher skin on the back and more sensitive skin on the belly.
Possibly, yeah. And then there's collagen.
Right. Collagen came up a lot in this study.
Yeah.
They found that collagen type 1 was dominant in the salmon skin.
Yeah, it's a major structural component.
So why is collagen so important for fish?
Well, it provides structural support and elasticity.
Okay.
And for fish, it's crucial for locomotion, swimming, and maintaining the integrity of that skin barrier we talked about.
And spatial transcriptomics let them pinpoint the locations of different collagen types?
Exactly. They could see exactly where each type was expressed, which
Ugh.
you can't do with traditional methods.
That's amazing.
Mm-hmm.
Okay, so let's talk about keratins.
Okay.
They found a ton of keratins in the epithelial tissue.
Yeah, keratins are like the building blocks of the outer layer of skin.
Right, so that makes sense.
But here's the thing. They found keratins expressed in non-epithelial cells in fish, too.
Wait, really?
Yeah, and that's different from what we see in land animals.
Ah, interesting.
So it suggests that keratins might have other roles in fish that we don't fully understand yet.
It's amazing how this one study is opening up so many new research avenues.
It really is. It's just scratching the surface.
So much to explore.
Yeah, it's fascinating.
Well, let's dive deeper.
Let's do it. It really is opening up new avenues, and they didn't stop at collagen and keratins.
Right.
They also looked at this protein called vimentin.
Mhm.
It's often found in cells that can turn into different types of tissue, like bone and cartilage.
And I bet spatial transcriptomics was useful here too.
Oh, absolutely.
Right.
They were able to confirm, the known distribution patterns of vimentin, which basically means,
So it worked.
yeah, it's like double-checking the map and confirming, like, yep, that's where it's supposed to be.
Adds another layer of confidence to their findings.
Yes.
So it's not just telling us what's there, but also where it is.
Exactly.
That's powerful stuff.
It is. And to make sure they were on the right track, they use these things called tissue-specific gene markers.
Okay.
Think of them like flags that signal, hey, this is epithelial tissue, or this is bone.
So it's like in a museum, they have those little labels next to the artifacts.
Yeah.
It helps you identify what you're looking at.
Exactly. And in this case...
Maybe give you some context.
Yeah, and in this case, it helps them identify different tissues in the skin and understand their specific functions.
And how they all fit together.
Exactly. They were also able to compare their findings to this huge microarray database called STARS.
STARS.
Yeah, it's like a massive collection of gene expression data from all sorts of tissues and species.
Wow. So they're putting their findings in a larger context.
Yeah, and guess what?
Right.
They found similarities between salmon skin gills and the olfactory rosette.
Okay, remind me what the olfactory rosette is again.
It's the part of the fish that allows them to smell.
Ah, okay, so we're talking about skin gills and the nose, essentially.
Yeah, basically. And these are all mucosal organs.
Mucosal organs.
Yeah, meaning they have that moist lining tissue.
Okay.
And the fact that they share similar gene expression patterns suggests...
That they're connected somehow?
Yeah, there's a link between their function and how their genes are expressed.
So it's like finding a common thread between these seemingly different parts of the fish.
Exactly.
And this also means that this technique could be applied to other species and tissues, right?
Absolutely. It opens up a whole world of possibilities.
Wow.
Especially for aquaculture. Imagine being able to identify, early warning signs of skin problems in fish.
Oh, that would be huge.
Yeah, or developing treatments that target the specific affected areas.
It would revolutionize fish health management.
It really could. We're talking about reducing antibiotic use,
Okay.
improving fish welfare, and making aquaculture more sustainable overall.
That's amazing.
Yeah.
But this research wasn't without its challenges, right?
Right.
I remember reading something about difficulties classifying all the transcripts.
Yeah, it was tricky to accurately classify, all the transcripts into distinct tissue clusters.
So like putting all the pieces of the puzzle in the right place.
Exactly. And it highlighted the need for even more advanced techniques,
Like what?
like combining spatial transcriptomics with single-cell RNA sequencing.
Okay.
That could give us an even more refined view, like down to the level of individual cells.
So even with these cutting-edge tools, there's always room for improvement.
Always. That's science. Always pushing the boundary.
Always more to learn.
Yeah, trying to develop even better tools to understand the complexity of life.
This is really cool stuff, and it sounds like we're on the verge of a big leap forward in understanding not just fish health,
Yeah,
but human health too.
absolutely.
Are there any specific findings from this study that point to those broader applications?
Oh, yeah, definitely. Remember those epithelial gene markers we talked about?
Mm-hmm.
Some of them have well-known functions in humans, too. Okay.
For example, they found high expression of claudin-1.
Claudin-1.
It's a protein that plays a crucial role in maintaining the integrity of our skin barrier.
So if claudin-1 isn't working properly, our skin can become more vulnerable to, irritation and infection.
Exactly.
I see how that would be relevant for a wide range of skin conditions.
Absolutely. And they also found myosin,
Myo.
which is involved in cell migration.
Okay.
And that's a crucial process for wound healing in both fish and humans.
So by understanding how myosin is regulated in fish skin, we could potentially gain insights into how to improve wound healing in humans.
Precisely. And they also identified another interesting gene called Kruppel-like factor 5-like.
Wow, that's a mouthful.
Yeah, the long one, but it's involved in cell growth and differentiation,
Okay.
which is essential for maintaining healthy skin and repairing damage.
It's like these fish are giving us a window into some fundamental processes that are shared across species. They really are. But what's even more exciting is that there are many other epithelial gene markers they identified whose functions are still unknown.
Oh, wow. So there's a whole new world of potential discoveries waiting to be made.
Absolutely. This study has laid the groundwork for a deeper dive into the complexities of fish skin and its implications for understanding skin health more broadly.
Well, this deep dive has certainly opened my eyes to the fascinating world of fish skin and the power of spatial transcriptomics. It's clear that this technology has the potential to revolutionize how we approach not just fish health, but human health as well.
I completely agree. And the beauty of scientific discovery is that we never know where it will lead us. What starts as a study on fish skin could unlock breakthroughs that have far-reaching implications for human health and beyond.
Thanks for joining us today on this deep dive into the world of Atlantic salmon skin and spatial transcriptomics.
It's been my pleasure.
We've learned about the cutting-edge techniques used to map gene activity in fish skin and the surprising connections between fish and human skin health.
It's fascinating stuff.
Who knew fish skin could be so interesting? Until next time, keep exploring the wonders of science.